Hey students, Mr. Lucero. Just wanted to create a recording of the lesson from this morning. If you remember, there were three objectives for this lesson. The first one was to talk about the major difference between DNA replication in prokaryotes and in eukaryotes. The second objective was to talk about the basic structure of a gene and the flow of information in a cell. And then third was to talk about that first process of transcription and how that process looks similar and different in prokaryotes and eukaryotes. So let's get started here by looking at DNA replication. So at this point, I hope you had a chance to look at this figure. This is a key figure from this unit that summarizes all of the key proteins that are involved at the site of replication, which we call the origin of replication, or ORI for short. And if you look at this diagram, we are actually looking at one side of this origin of replication and kind of that where those strands are being separated and where that leading strand is being synthesized towards. That's called the replication fork. And then, of course, we've got the lagging strand that's synthesized in Okazaki fragments. And the reason I wanted to talk about this is because this idea of an origin of replication, right, the site at which DNA replication begins, is kind of at the center of the difference between DNA replication and prokaryotes and eukaryotes, right? If you remember, right, a prokaryote is different from a eukaryote in that does not have a nucleus or any membrane-bound organelles, for that matter. So like mitochondria, chloroplasts, Golgi, right? Those are only found in eukaryotes. And if you also remember, right, prokaryotes, they have circular DNA. They actually have one large circular chromosome. And eukaryotes actually have linear chromosomes. They have 23, well, in the case of humans, right, 23 linear pairs of chromosomes. And not only that, right, there's far more DNA usually in a eukaryote than it is in a prokaryote. And so when DNA replication occurs in a prokaryote, there's actually only one origin of replication found on that circular chromosome. So this is representing the origin of replication seen in a prokaryotic cell, such as an E. coli cell. And as you look at this circular chromosome from this prokaryote, there's only one origin of replication, and we see, right, on both sides of that origin of replication, right, the replication fork is going in both directions. Okay, but in eukaryotes, a little bit different because there's so much more DNA and that the DNA is not one continuous strand. We actually have multiple origins of replication. There are lots of different origin replications that start DNA replication simultaneously across the genome, allowing for more efficient replication of the entire genome. All right, so key difference, multiple ORIs in eukaryotes and just a single ORI in prokaryotes. Okay. Now, I also mentioned at this point that if you remember, a key part of DNA replication, it has to do with directionality. This is the key reason why the leading strand and the lagging strand have to be synthesized differently. And that has to do with the fact that, if you remember here, right, DNA has directionality, and that refers to the 5' and the 3' ends of the strands, with the 5' end representing the end where you will find that 5' carbon on the sugar attached to the phosphate, which is right here. But on the other side, we have the 3' carbon that's attached to the hydroxyl, or OH, right? And in a DNA molecule, that has two strands, we say that it is anti-parallel because one strand runs from 5 to 3 and the other runs 3 to 5. And whenever a new strand of DNA is being synthesized, such as in semi-conservative DNA replication, that new strand can only be synthesized in the 5' to 3' direction. And what that means is that new nucleotides, right, the monomers of this polymer that's being formed, can only be attached to that 3' end of the new strand, right? So it's going to elongate in the 5' to 3' direction. And this is true whether you are synthesizing DNA in DNA replication, and also when you're synthesizing RNA, which we're going to talk about in transcription. Those polymers can only be formed in the 5' to 3' direction. Okay, so let's talk about our second objective for this lesson, which has to do with the structure of a gene and the main information flow that occurs within a cell. So let's start with actually that second piece, right? So whenever we describe the flow of information in a cell, what that's referring to is that in a cell that contains DNA, right, that DNA is split up into different segments that are called genes, right? And each of those genes stores information in the form of a specific order of nucleotides that are used to direct the synthesis of a particular type of protein, right? And this diagram actually represents how that's done. So this is the main way that information flows in a cell. This is also referred to as the central dogma of biology, since it's seen across the board in all organisms as how DNA controls the production of proteins, which do all of these things in organisms. Okay, so let's do a quick summary of this central dogma. The idea is that if you have a gene, such as gene 3 here, that stores information to make a particular protein, that DNA can be used as a template to be transcribed through the process of transcription into an mRNA molecule, that then can be translated into a protein, all right? And if you remember, right, the building blocks of a protein are amino acids. There are different types of amino acids that have different properties, and so the specific order of amino acids will dictate how the secondary, tertiary, and quaternary structure of the protein folds, right? And that ultimately determines the function of that protein. Now, just a couple other things to note that we talked a little bit about in Unit 1 are these base pairing rules, right? So if you remember, if we look at the DNA strand, on the interior of this DNA molecule, the nitrogenous bases pair in a particular way. Adenine always pairs with thymine, and guanine always pairs with cytosine. If you remember, adenine and guanine are purines, and thymine and cytosine are pyrimidines. So in other words, a purine is binding with a specific pyrimidine in a very predictable way. The only difference is when we talk about RNA, right, that those base pairing rules are slightly different. RNA does not contain thymine, it contains uracil. And so those base pairing rules, instead of it being adenine pairing with thymine, it'll be a uracil in RNA, but guanine will still pair with cytosine. And so you can see the link between this mRNA molecule and the DNA template. In fact, the top strand was used as the template for this mRNA, right? We see A is pairing with U, and C is pairing with G, and C is pairing with G, and so on and so forth. So that is just a quick refresher on how bases pair together in DNA and in RNA. Okay, and you also might remember from Unit 1, but also from your bio class, that this mRNA is translated from the language of nucleotides into the language of proteins or amino acids by being read in groups of 3 letters that we refer to as a codon that code for a specific amino acid. So this codon UGG codes for tryptophan, a particular amino acid, and UUU codes for phenylalanine, and so on and so forth. And so you get a polypeptide that folds into its specific shape. Okay. Now, transcription, as you can see right here, which we're going to focus on for the remainder of the lesson, right, is this process of using DNA, a gene, as a template to make an mRNA molecule. And I want to show you here in this diagram that the enzyme that performs transcription is called RNA polymerase, or RNAP for short. And it's this pretty amazing enzyme that actually kind of simultaneously acts as a helicase and a polymerase that unwinds the DNA and then also helps synthesize the mRNA polymer based on the nucleotide order of the DNA. And you also notice, right, that look at the mRNA that's being synthesized. You notice the direction, right? So this mRNA is being synthesized from 5 to 3. Those new nucleotides are being added to the 3' end of that growing mRNA strand, right? So just like in DNA replication, RNA is made in the same direction. Okay. So let's talk a little bit about the basic structure of a gene, mainly because as we get more in depth into transcription, and later in this unit when we talk about how gene expression, in other words, when a gene is transcribed and translated, we see that gene is being expressed. We're going to talk about how that process is regulated. And so we have to know a little bit about the anatomy or the structure of a gene, this section of DNA. What are some of the components that make it up? So if we were to take, for example, gene 1, and we were to zoom in, we can identify a couple of components of this section of DNA, and these components are shared by every gene. I'm going to walk through those components here. So this this black line is just going to represent this stretch of DNA, and we're going to use different colors to indicate the different sections of this gene. So at the kind of center point of this stretch of DNA called a gene, we have a section known as the coding region, right? So the coding region is the section of the gene that will directly code for amino acids, right? This is going to be the section of the gene that is transcribed and translated into protein, ultimately to make that specific order of amino acids, right? So this will become the mRNA and then the amino acids, right? That's the role of the coding region. In other words, that's the main location of the information needed to make the protein. However, that's not all that a gene is made up of. There are other sequences that are found within the stretch of DNA that contribute to the gene's function. And so the first one I'm going to talk about happens right before, or is seen right before the coding region, and we call this the promoter. All right, the promoter is a sequence of DNA where RNA polymerase attaches to initiate transcription. So RNA polymerase, which is a protein, will attach to specific regions of the promoter before proceeding with transcription, right? Which makes sense, right? The RNA polymerase has to be able to attach to the DNA so then it can perform this function that we see in this figure above. Okay. Now we also have at the very end of the gene we have a section that's referred to as the terminator in prokaryotes, or just the terminating sequence in eukaryotes. The mechanism is slightly different, but the major function that at least we need to be familiar with is that this is where RNA polymerase detaches, right, once transcription has completed. Okay, so we've got a promoter region, the coding region, the terminator or terminating sequence region, and then the last region that we're going to especially see in eukaryotes, and we'll talk more in depth about this later in Unit 6, is right here. And these are referred to as enhancer regions. And these sequences are key in determining when and where in an organism a gene is expressed. And this will be where specific types of proteins called transcription factors, which we'll talk about later in Unit 6, where they connect to, just like RNA polymerase binds to the promoter, and they help regulate when and where this particular gene is expressed. Okay, so these elements all make up what we refer to as a gene. So a gene is not just the coding region, it includes the enhancer, the promoter, and the terminating sequence as well. Now, two terms that I referenced in this lesson are upstream and downstream. So just for terminology, right? So when you hear the term upstream, that just means you're referring to before the start of usually the coding region. So if I said the promoter is upstream from the coding region, that's what that means, is that it's occurring prior to. Downstream will be the opposite, right? So the terminating sequence is downstream of the coding region, and the promoter region and the enhancer region, they're downstream. Okay. So that's the basic structure of a gene, and we're going to use these different components in the rest of this lesson, but also as we talk about regulation as well. Okay, so let's talk about the last objective of this lesson. I want to talk a little bit more in depth about how transcription looks similar and different in prokaryotes and eukaryotes. So what we know so far, right, is that transcription is where RNA polymerase attaches to the promoter and makes an mRNA transcript of the coding region based on the specific base pairing rules of A pairing with U and G pairing with C. However, when we think about prokaryotes and eukaryotes, these processes, while the end results is shared, kind of the details of the process are a little bit different in those two categories of organisms. So to remind you, right, a eukaryotic cell that looks like one of these right here has a nucleus. This is where transcription occurs, is in the nucleus. That makes sense, that's where the DNA is found. But a prokaryotic cell, such as this bacteria cell, does not have a nucleus, and so transcription is just occurring within the bacteria, not separated from the rest of the cell. Okay, and that kind of gets at the first key difference between eukaryotes and prokaryotes when it comes to transcription, is that there is separation of transcription and translation in eukaryotes, but not in prokaryotes. Right, and we can see that in these diagrams over here, right? Because transcription is occurring in the nucleus, it kind of separates it from the process of translation, which occurs outside the nucleus in the cytoplasm. But in a prokaryotic cell, that separation doesn't exist. And so what that means is that actually in prokaryotic cells, transcription and translation actually occur simultaneously. And we can see that here in this diagram, that actually this is a prokaryotic cell performing transcription. Here we see RNA polymerase proceeding through the DNA, creating this mRNA. And at the same time that that's being created, ribosomes, which we'll talk about Friday, actually start to attach before the mRNA has been completed, and will start to translate it into the polypeptide. And so there's this simultaneous transcription and translation in prokaryotic cells simply because there's no separation. So that's our first key difference. The second difference is that in eukaryotes, we have something called the transcription initiation complex that is heavily involved in this very complex regulation of gene expression that's not seen in prokaryotic cells. Now we're going to talk about this transcription initiation complex next week, but this figure right here kind of summarizes it. So you'll see some elements that we just talked about. So we have the enhancer, we have the promoter, and then we have gene A, which in this case is referring to the coding region of this gene A. Okay, and then you'll notice that you've got different proteins kind of all clumped together. And collectively, this clump is known as the transcription initiation complex. So you've got RNA polymerase attached to the promoter. You've got all of these different transcription factors and mediator proteins that are attached to these enhancer regions that are ultimately going to allow for more complex regulation of gene expression in eukaryotes. But again, this is not seen in prokaryotes. So that is another difference between prokaryotes and eukaryotes. The third difference, though, is that there are different termination mechanisms in prokaryotes and eukaryotes. So you might have noticed right on the previous slide that I mentioned how in prokaryotes, typically it's a sequence known as a terminator at the end of the coding region that causes RNA polymerase to detach. But in eukaryotes, there's a slightly different mechanism that has a terminating sequence that ultimately results in the same thing, but it looks a little bit different. And so that is another difference between prokaryotes and eukaryotes. It's not important that you know what those mechanisms are, just know that that is different between prokaryotes and eukaryotes. Okay, but then lastly, one of the major differences between prokaryotes and eukaryotes in terms of transcription are that in eukaryotes there is something called mRNA processing in eukaryotes. And this is possible because of this separation in the nucleus. There's a lot of RNA processing that occurs prior to translation, but in prokaryotes, there's not really time for this because translation just starts immediately. There are kind of three major things that happen in RNA processing that we're going to go through. So the first is something called a 5' cap is added to the mRNA. The second is a poly-A tail. The polyadenylation tail is added to the 3' end. And then lastly, a process called splicing or alternative splicing occurs to the primary RNA transcript. I'm going to walk through these three things in the next portion. Okay, so let's talk about splicing. Okay, so this diagram is going to help us to outline this process of splicing. Just for the purposes of this class, pay attention to the things that we are familiar with in terms of the structure of the gene. So we have the enhancers that are upstream. We have the promoter. We have the coding region. This is actually from a eukaryotic cell, and so the terminating sequence would be here. It's not labeled on this figure, though. Okay. And what I want to point out here is that if this is our gene, this is DNA, so this is our gene, right? So when transcription occurs, the immediate result, the product of that transcription, is something called the initial transcript. It's also called pre-mRNA. And what happens when that initial transcript is made are the first two RNA processing steps. The first thing is there is a cap that's added to the 5' end, and the second is a whole bunch of A's are added to the 3' end. Although only a few are included in this diagram, usually it's in the order of 50 to, in some cases, over 100 A's, adenines, are added at the end of the transcript. And there are really kind of two purposes, or three purposes, for the 5' cap and the 3' tail. The first is that the 5' cap and the poly A tail, they actually provide some protection from degradation in the cell. When this mRNA molecule exits the nucleus and enters the cytoplasm, there are all of these enzymes that will begin to degrade the mRNA. And so this 5' cap and poly A tail just provides a little bit of protection to the information stored in the main part of this mRNA, which is the information to make the protein. So it provides some protection. The second is that it helps with this mRNA exiting the nucleus. There's this process whereby this mRNA needs to leave through a nuclear pore so that it can enter into the cytoplasm or the rough ER to be translated, and that 5' cap especially helps with that process. And then the last reason is that especially the 5' cap and 3' poly A tail are going to facilitate the ribosome attachment for translation. Okay. All right, so there are our three main purposes. So let's talk a little bit more about the other element of RNA processing, which is splicing, and that's the major component that we see in this diagram. So once the pre-mRNA has been modified with that 5' cap and the poly A tail, you'll notice that splicing refers to the removal of introns, right? Within the coding region, we have these sections called introns and exons. And just for this class, an intron and an exon, they're differentiated based on the fact that exons directly code for amino acids, but introns do not code for amino acids. All right, so the information that codes for specific amino acids are found in the exons, not in the introns. Side note, there's been a whole bunch of research into the purpose of introns. They were initially coined as junk DNA, but it's now been found that they actually have a lot more purpose than initially thought. But at least when talking about RNA processing, those introns get spliced out. They get spliced out in this last step of RNA processing. And so as you'll see, right, we've got exon 1, 2, and 3, the green portions of the coding region, and the blue intron 1 and 2 are removed from that pre-mRNA transcript. And the exons, 1, 2, and 3, are joined together. And so we have our final mRNA that then can exit the nucleus and be translated. Okay, so that's splicing. That's the last step of RNA processing. Now, there is one more component of splicing that we talked about this morning that contributes greatly to eukaryotic complexity. Remember that this process of RNA processing, splicing, this is happening in eukaryotes only, right? There's actually something called alternative splicing that I'm going to talk about here in just a moment that, like I mentioned, helps contribute a lot of complexity for eukaryotic organisms. Okay, so it's a very similar figure to help explain this idea of alternative splicing that's only happening in eukaryotes. Just a quick overview of this figure. We only have a coding region represented in this gene. They don't show the promoter or the terminating sequence. But we've got 6 exons, my apologies, in this coding region and 5 introns. Right, and we've got our primary RNA transcript, right? We've got the 5' cap, the poly A tail. But then when it comes to splicing, many eukaryotic organisms have this ability to alternatively splice different exons together. Let me tell you what I mean here. Let's look at this first one. Right, so this is just a regular, or this is an example of alternative splicing, because if you notice, which exon has been left out? Well, exon 3, right? We've got 1, 2, 4, 5, and 6. Exon 3 has been left out as those introns were being spliced, right? They took out not only the introns, but also exon 3. Looking further at number 2, in this case, we've removed 2 exons, exon 2 and exon 4. And then in this last one, we're missing exon number 2 only. Why is that important? Well, so it's important because the result of alternatively splicing different combinations of exons together means that you are going to get 3 different proteins from the same gene. And this allows, like I mentioned, for a eukaryotic organism to have an immense amount of complexity because the number of genes that their genome contains does not correspond to the number of proteins and the functions that those proteins perform in the organism. So humans, for example, I think have about 25,000 genes, but have over 100,000 different types of proteins that perform all these different functions that make us very complex. And so alternative splicing is behind that complexity. Okay, so I hope if you missed anything from this morning that this helped to clarify. Please reach out if you have any questions, and have a good rest of your afternoon.